Andretti, and I'm going to tell you about this work we have done recently. Most of the time, cells maintain a huge gradient of calcium concentration across the plasma membrane, and in this process, the plasma membrane calcium ATPase, or PMCA, has a critical role. We know that PMCA has three domains, P, N, A, and a transmembrane segment inside the lipid bilayer. We don't know exactly how PMCA transports calcium, but PMCA has a sister, the sarcoplasmic reticulum calcium pump, or SERCA. And we know a lot about SERCA function by recent crystallographic studies. We know that in E1 conformation, SERCA binds calcium and phosphorylates from ATP, generating the E1P intermediate. This intermediate relaxes to E2P and the calcium is released in the other side of the membrane. Finally, the SERCA desphosphorylates, starting the cycle again. Part of this information was obtained by using metal fluorides complexes that stabilize the SERCA in conformations identical to the phosphorylated one, and allow researchers to take snapshots of the protein in the phosphorylated intermediates. Metal fluorides mimic pretty much a phosphate in different states of the dephosphorylation reaction. Barium fluoride mimics the ground state, aluminum fluoride mimics the transition state, and magnesium fluoride mimics the product state of the dephosphorylation reaction of proteins. So our final goal is to understand the conformational changes involved in calcium transport in a PMCA. In this work, we aim to investigate if metal fluorides stabilize a conformation similar to the phosphorylated one in the PMCA. We wanted to study this conformation using a fluorescent probe, and we wanted to study how metal fluorides bind to the PMCA. To know if metal fluorides stabilize a conformation in PMCA, we measure the protein transport activity. We can do this by quantifying the phosphate released by the protein while transporting calcium. So look at the experiment. In the absence of metal fluorides, the PMCA has an activity, but when we added barium fluoride, we detected a decrease in protein activity. This suggests that when metal fluorides bind to the PMCA, they fix it in a conformation and can no longer hydrolyze ATP. We performed this experiment for different barium fluoride concentrations and also for aluminum and magnesium fluoride. These results indicate that metal fluorides fix the PMCA in a conformation, and this conformation is probably similar to the phosphorylated one. Then, we studied the location of the protein that binds ATP using a fluorescent molecule called eosin. In this work, we found that eosin binds to the ATP binding site and increases its fluorescence. If we measure the fluorescence of eosin at different concentrations, we detect an increase in fluorescence until the behavior saturates, because there is a limited number of ATP binding sites to which eosin can bind. We made the same experiment, but in this case, the PMCA was previously incubated with barium fluoride. In this case, eosin binds to the protein and increases its fluorescence, but the increase in fluorescence was smaller. This indicates that eosin has less fluorescence when bound to the PMCA in the phosphorylated state. This is probably because the ATP binding site is more hydrophilic in the phosphorylated state. And we saw a similar behavior for aluminum and magnesium fluoride. Then we measured the fluorescence of eosin over time. By doing this experiment, we could measure the speed by which barium fluoride binds to the PMCA. And we saw that the binding of barium fluoride to the PMCA is very slow. We performed this experiment for different barium fluoride concentrations and also for aluminum and magnesium fluoride. Based on this data, we could propose a model of how metal fluorides bind to the PMCA. In this model, PMCA can bind metal fluorides, but only in a state that is very infrequent, and this explains why the binding of metal fluorides is slow. However, when metal fluorides bind to the protein, they form stable complexes. So, we can conclude that metal fluorides bind to the PMCA, stabilizing the protein in a conformation and inhibiting the activity. The ATP binding site of PMCA is more hydrophilic in the phosphorylated state, and metal fluorides bind to the PMCA slowly, but they form very stable complexes with the protein. This information will help us in the future to gain more insight about the structure of the PMCA in the phosphorylated intermediate.